I'd like to welcome Filio Billia, who is the Director of Research at the Peter Munk Cardiac Centre. Filio has done important work, especially in the area of reactive oxygen species and the damage that induces in heart failure and the related signaling pathways. She is also the Medical Director of the Mechanical Circulatory Support Program at UHN, which is growing dramatically, and the co-director of the Cardiovascular Biobank at the Peter Munk Centre. She's going to talk to us this morning about molecular mechanisms in cardiomyopathy from mice to men. Phyllis.
Thank you. I'd like to take this opportunity to thank the organizers who asked me to give this talk, and also to say, why on earth did you give me this title? Because it is very overwhelming, as you will see, and what I've tried to do is not to talk about the 20,000 papers that have been published since the 1950s, but to give you a glimpse of some of the things that have been done and what the limitations are, and maybe open up thinking about where we could potentially be going with this, and maybe mice are not men, which I think is obvious. So I have no disclosures. Now, their heart failure, as Peter introduced me in terms of the many hats that I wear, I look after heart failure patients on a day-to-day basis, and I have to say it's overwhelming. It is, each patient is unique. Each patient has its own constellation or their own constellations of symptoms. And, it's a complex syndrome. The etiologies that causes heart failure are very diverse, from ischemic heart disease to peripartin cardiomyopathy to inherited problems as well. And while the definition is pretty straightforward in that the heart is unable to meet the demands of the body, it's actually a really complex syndrome. And I think having a unified approach is limited, and we need to really work together from all the three sites to really tackle this very complex problem. It is a huge problem. The impact, I know Mansour did give some numbers, based on our own numbers here in Canada, we're getting closer to the 1 million patients that are afflicted with heart failure, and 50,000 new cases every year of patients that are getting this diagnosis. And still the mortality is quite high in this patient population. And if you look at median survival once they're diagnosed, it's 1 to 3 years depending on your gender. But a 5-year mortality is very close to 50%. And as we age, as you can see from this terrible aging population, as the patient population gets older and older, the prevalence of heart failure also gets higher and higher. So it is an epidemic, and we've been limited in terms of what's been happening from a novel therapeutic perspective in the last 20 years, although I've had some exciting developments in the last 6 months with some interesting work, and which I'd like to talk about. And the hospitalizations along with that have been increasing. Now, let's talk about mechanism. So if you go back to the early reports, this is all electronic available information, not what you can get if you go and look at Osler's or initial descriptions of people back in the 1890s, etc. So just searching on PubMed, and you just search mechanism in heart failure, you get greater than 10,000 papers. If you broaden that and look at the pathophysiology, it's more than 20,000 papers, and that's very daunting because every part of that work has led us down a particular pathway to have a better understanding of this very complex syndrome. And some of the initial descriptions are from the late 1940s and 50s in PubMed actually look at the physiological considerations. Guyton, who actually went on to do multiple textbooks in physiology, actually published a really interesting paper looking at compensation and decompensation in acute cardiac failure, and some of the other things that were there, and things that we don't use anymore, like mercuritic diuretics, were really big in the 1940s and 50s, but that has really dramatically changed as we've developed a better understanding of what's going on. But the molecular papers didn't start to come out until the late 80s. So here's one paper by Victor Zhao. So Victor Zhao is a very preeminent, very important man in this whole area of understanding hypertension, heart failure. And I'll put in a ploy for some of the students in the audience, because he will be here in Toronto in a week's time. The Institute of Medical Science, which is a graduate department, is having a research scientific day, And Victor Zhao is the keynote speaker for that, and it would be an amazing opportunity to hear this man talk because he's been in the business for 40 years, if not longer, and is one of the preeminent scientists in this field. But when you look at what's been done early on, we're talking about in situ hybridization as a marker of understanding what's being expressed, as well as doing just simple northerns. We are so far away from that, but that's where the work started, and it took molecular biology as it became revolutionized with polymerase chain reaction and all the other things that have been happening in the 90s and the early 2000s have really changed what we've been able to do. And if we look, in my mind, I think there are two different roads in thinking about the molecular mechanisms of heart failure. If we think about this in two ways, one is the genetic predisposition that really falls into what Simo has been talking about and what Dr. Watkins has been talking about, which is inherited forms of cardiomyopathy, such as hypertrophic cardiomyopathy, the familial forms, ARVC, and restrictive. I think that's different in some ways, although similar in others, but we're looking at genes that are predisposing to those types and forms of cardiomyopathy as opposed to understanding why the cardiomyocyte fails or the heart fails as an organ that is not related to a genetic predisposition. And that's a much more complicated, I think, avenue. And our current understanding of these mechanisms really relies on the genetic modeling that has happened in the mice. And the revolutionary advantage has been since the sequencing of the human genome as well as the mouse genome, we're able to manipulate the mouse genome at a level that we cannot do in larger animals. We can do it in such elegant manners that we can actually knock out a single gene and then see what the consequences are of that gene. But if we look at the hallmark features of heart failure at a morphologic and functional pathway, hallmark features include pathological hypertrophy in the early stages, where we see pathological hypertrophy being actually compensatory and then becomes decompensatory. And then we see, as a result, an increase in the heart size, and over time we see a decrease in heart function. This is associated with fibrosis and apoptosis of the cardiomyocytes. We see increased expression of a fetal gene program, so there's an alteration in the gene expression profile. And also associated with that is the inability to handle calcium in a way that is normal. In addition to that, we have inflammation, and we have a whole bunch of other things which I think should be included, which is not only changes in the cardiomyocytes themselves morphologically, but at a cellular level is the fact that we need to think about oxidative stress. This is cytoplasmic versus mitochondrial changes in metabolism and energetics that happens within the cardiomyocytes. And then in the system as a whole, the overactivation of neurohormones and the receptors, as well as epigenetic changes that can completely alter everything. Now, this is supposed to be overwhelming. I did it on purpose because now this is sort of a signaling cascade that has been described in a recent, well, this is not so recent, but it's actually a really nice figure to show because if we go more recent, it gets more and more complicated. But this is a signaling cascade from the level of the membrane down to the nucleus at the bottom where we have activation of the transcription factors. So this is signaling. You can easily show representations for this, for what happens within the mitochondria, what happens with energy metabolism and the alterations. And the reason I show this is you could pick any one of these targets and probably someone has knocked it out in some shape or form in a mouse and looked at it in isolation. And what I've done here to show you an example of this is I've actually circled GSK3 beta because it's a recent publication in Circulation Research in April of this year as an example. Not to pick on this publication, but to show you that in isolation, this is what we are all not capable of doing, but actually what we all have been doing, and whether it's enough is what remains to be answered. So this is an elegant study that was done by Thomas Force's lab, and this is just to show you that GSK3 beta is a signaling molecule that's actually present in two isoforms, alpha and beta. Some of the initial mice were actually produced here in Toronto by Jim Woodgett, And I remember, because when I was doing my PhD, I had a master student that was analyzing both of these mice in isolation, and unfortunately when you do that, one form of the isoforms actually takes over for the function of the other, so they didn't actually get a very dramatic phenotype in the mice. It wasn't until now, we're talking probably more than 10 to 15 years later, when the double knockout came out, that you start to see that there is a phenotype that you can see in isolation in the heart. So this is a cardiac specific knockout of both alpha and beta to give you a dramatic phenotype. And what I'm trying to show you here is some of the work that we all do. Anyone who has a basic science lab and does any knockout technology will knock out a gene, we'll see what it does in the heart. We'll look to show you that the expression has been reduced effectively when you give tamoxifen in an inducible model. We show you nice, pretty pictures of what the heart looks like. It dilates, it thins over time. We show you that there's a drop in fractional shortening there at the bottom left, so that they develop a functional cardiomyopathy, and then there's fibrosis, which is associated with it, which is at the bottom right. So these are supposed to be classical features of heart failure. And then we end off the paper with a nice model. And we all are guilty of this because papers want this. They want you to do a really extensive workout. Some of us do microarray work to really isolate and think about the mechanism. And then we find in the end result is figure 6 or 7. Down at the bottom, you have this beautiful model, and this is what you propose as the mechanism. So it's great. In this example, what he's been able to show, the lab has been able to show, is the double knockout actually goes through multiple rounds of DNA synthesis without actually exiting the cell cycle to actually then go through cytokinesis, which is when two daughter cells then form. So you have not just binucleated cells, but you have 4 or 5 nuclei within a cardiomyocyte, and that then leads to the phenotype and increased levels of apoptosis and what he's calling cytokinetic catastrophe. So if we go back to this and think about that was one gene in isolation, and if you do all of that work and you try to do all of these in isolation, it's putting the whole bigger picture together, which is quite difficult to do. And if you try to do it through a microarray analysis, then you get the criticism that you haven't proven the mechanism, and that is all suggestive but not mechanistic. So it's a bit of a vicious cycle, because then you have to go back and do all that mechanistic work to prove. So you're catching your tail back and forth. If we look at this cell a little bit differently, and what I'm trying to show you here is some of the new treatments that have come out from a heart failure perspective that we're now using in the clinic, and then seeing how from outside of the cell and the neuroactivation pathways that are affected and how they lead into the cell. So we've worked out mechanisms within the cell, and we've developed some interesting new drugs that have been on the market for a very long time, and a very new player, LC2696. Which is a new player, and it's a dual player. We've also been using ACE inhibitors for a very long time, which actually is involved in inhibiting the conversion of angiotensin 1 to angiotensin 2. And dobutamine and milrinone are commonly used in very, very sick patients, and also all that work has been done in dog models back in the early or mid-'80s, and all the cascade work has been done either in vitro or also been done in whole heart preparations in mice, rats, or dogs. So what I wanted to do is just to highlight that there is more to this game, but the thing is, what hasn't really happened for us, aside from LCZ696 and maybe ivabradine, which is another new drug that's yet not available in Canada, soon to be, is that we've kind of hit a stagnation point except for this one drug. And if we look at what's been happening over the ages, so we started off 1950s using the mercurial diuretics, then we've moved our way in the 1990s with the exploration of these new, what we thought was new drugs, ACE inhibitors, beta blockers, which are the mainstay of our treatment for patients. And then we've had to go into a mechanical era, because we've had reached a stagnation point in terms of how improving survival with our patients and what are we able to do. So in places like ourselves, we are able to actually offer some degree of transplantation, again, not enough for the demand, and having mechanical circulatory support program, we can support some of our patients, again, not enough for the demand. So we have had a bit of a stagnation, and hopefully what we can do is use our knowledge to devise novel therapies going forward. But the question is, are mice the appropriate model? The reason I bring this up is a lot of the drugs that have been used up to date and have been developed and have been proved to be efficacious, for the most part, are there to treat systolic heart failure. And this is a nice, I think, representation of what systolic heart failure looks like, which is you have a large heart that dilates and thins out, usually after a myocardial infarction. That's the most common inciting event, as opposed to diastolic heart failure, which is a near normal or smaller chamber size, a normal ejection fraction, but a thickened wall, so its inability is in relaxation. And this is more commonly in our elderly, our diabetics, and our hypertensives. And that while they are different in mechanism, the outcomes are still bad for diastolic heart failure as they are for systolic heart failure. And I think while it's not as sexy to work on diastolic heart failure, it is actually just as important a clinical problem. And a lot of the drugs that have been developed and based on animal modeling has been, the ones that have been successful thus far have been for systolic. Let's go back to LC2696. It's one of these most promising developments that has happened in the last year. This is from a review at the end of last year in 2015, which shows you the mechanism. It is an angiotensin receptor blocker with a Neprilysin inhibitor, and what it does is has dual actions on inhibiting the renin-angiotensin system while augmenting the favorable effects of the natriuretic peptides. And the trial that was initially published in 2014 in the New England Journal of Medicine, the Paradigm Heart Failure trial, actually was ended early because of the overwhelming benefit in using this agent, and the numbers needed to treat were as low as 21. So a very exciting drug, with definitely the basis of the work being from animal models. Some of the newer, not newer necessarily, I shouldn't say newer, because LCZ is a little bit newer than Circa, but another way of thinking about this and another avenue is to look at another problem, which is the abnormal handling of calcium. And using a targeted approach to go after this, because calcium is integral in the involvement of contractility, excitation, contraction, coupling. And Circa-2 has been shown to be responsible for the calcium reuptake during this process. And the animal studies, as well as in human failing myocardium, do show that in the failing heart, whether it's animal or human tissue, you do see a reduced Circa-2 activity. And where this work has gone forward and is actually pretty exciting is there are now gene therapy trials looking at restoring Circa-2 levels in cardiomyocytes using the adeno-associated virus. And this is just to show you some of the trials that are underway and are near completion, actually, if not completely completed. Cupid-2 have been looked at phase 1, phase 2, as well as phase 2B, and up to 250 patients. So we're still looking at small numbers of patients. And then for Circa in particularly, we're delivering this in an intercoronary fashion. And one prior pilot study was done actually in LVAD patients from a safety perspective, and having some really dramatic, interesting results. But I think a lot of the work that has been done to date shows that while mice are a good starting point in terms of helping us understand this cascade of potential events that could be happening within a cardiomyocyte or within a whole organ, mice are not men. And what we have to think about is the 60 years of literature that have been out there, and the many hours and a lot of money that has been put in to try to look at this in the failing heart. And yet we've been limited in what we've been able to develop in the last 20 years. And the current understanding may have been, our starting point may have been the mouse, but these models may not or do not always recapitulate the human cardiomyocyte phenotype. So getting human tissue is a problem, right? No one's going to donate their own heart. No one's going to allow you to take extra biopsies, or very rarely can you get, and sometimes biopsy tissues is tiny, it's a flake of tissue and you can't really do a lot with it. Limited survival, let's say you do get a source of human primary cardiomyocytes. They don't last long in culture. They're not a renewable source. So we need an alternative, and what is the potential for that? And to be honest, I've certainly gotten interested in this avenue in the last year or so, but I think using the technology of human-induced pluripotent stem cells may provide us with an avenue for that. And we should be thankful to the Yamanaka group back in 2006 that was able to show the initial work of this. The original paper described in Cell, and then the ability to actually reprogram these cells into cardiomyocytes. There's a couple of, I think, competing groups that were doing this at about the same time in 2008, but I just decided to show the paper from Dr. McClellan's group. So there is the ability to take cells from either the peripheral blood or skin, biopsy, reprogram them into induced pluripotent stem cells, and then reprogram them. And there have been this is a potential resource for human cardiomyocytes that can be disease-specific. So you can take specific populations of patients, generate disease-specific cardiomyocytes from these, but there have been some concerns raised about these cells. And the thing is, do they recapitulate the tissue? How should they be optimally grown? A 2D versus a 3D structure? What is going to be your gold standard as a surrogate and a comparator? The maturity of these cardiomyocytes is also in question, and do they mature enough, and how can you mature them in culture? Does putting them on, for example, a cardiac wire and stretching them help to mature them? So there's a lot of unknowns, but there's a lot of potential, I think, in this field going forward. Using drug development for this is an interesting approach because it can be done in patients in developing a personalized medicine approach. You can do drug toxicity trials as well with pharma, and then you can actually do the similar kinds of things that were done in mice, but do them from human primary cells, reprogrammed cells, to see can you recapitulate what you think is going on in the patient and compared to what you see in the dish. So there is still some concerns on many levels. Does this fully model human cardiovascular disease? Now, there have been many reports, I'd say in the last 5 years or less, that have shown the proof of principle for disease modeling, and I've listed here a few things. One thing I didn't include here was actually POEM syndrome as well. HCM, ARVC, I think those are clear winners here. Cardiotoxicity, what patients develop cardiotoxicity, familial dilated cardiomyopathies with troponin T and LMNA mutations in particular. Long QT has been very successful and very exciting. And in our own institution, we have a lot of interest from the arrhythmogenic side of things as well. This is some of the data that's been published from the various groups looking at various gene mutation-specific mutations and listing them, just as an aside from a recent review at the end of the year in cardiac failure. So we have things as troponin T, lamin A, myosin heavy chain for hypertrophic cardiomyopathy, or depending, right, restrictive cardiomyopathy, depending on the actual mutation. So in conclusion, I think mice have provided us an incredible resource for understanding the potential cascade of what can happen within a cardiomyocyte or within an organ and within the body, actually. But I do think that we have limitations and that there's lots of evidence to show that they don't necessarily recapitulate. And when we try to go to larger animal models or even to human clinical trials, what we find in animals does not actually work in man. So the breakthroughs have been huge, though, but we need maybe to think about larger animal models as a surrogate, or probably even the IPS world and differentiated cardiomyocytes that are derived with them as an experimental alternative to this approach. Or either not necessarily in isolation, it could be as a complement to elucidate mechanisms in a patient-specific